All animals have a very similar set of basic genes, and yet we're so different. So how do you explain that? What's the answer?
The answer to this mystery will take Will Wright back to the beginnings of the toolkit, to some of the earliest moments in creature making, and to one of the strangest ancestors we have on the evolutionary tree. Nothing can match evolution's awesome power for creating creatures. While designing a new game that simulates evolution, Will Wright is investigating what the latest science has uncovered about evolution's creature making. A toolkit of genes that shapes the intricate details of size, structure, pattern, and parts in all animals. How did the toolkit get set in motion in the first place? To find out, Will Wright is retracing its steps. From our present-day body design, we're headed to one of its earliest defining moments, when the toolkit was deployed in our earliest ancestor, a worm. The Earth has gone through a number of particularly intense bouts of creature making, but none as forceful as a time some 530 million years or so ago. It's known as the Cambrian explosion. A burst of animal forms appeared, the ancient ancestors of virtually all the animals that swim, fly, and crawl today. Is this the moment when the toolkit took off?
What's that?
Oh, it's a carnivorous terror bird from South America. But I actually have something a lot older to show you.
Okay.
Will Wright is visiting paleontologist Neil Shubin to find out how toolkit genes played a role in expanding evolution's creativity.
So here's where creature making went crazy. I mean, back about 530 or so million years ago. You know, you see just a blizzard of diversity of creatures coming about.
This is the Cambrian explosion?
Exactly, yeah, exactly. You start before the Cambrian, at about 600 million years ago, with creatures like this. Doesn't look like much. It's just a disk.
Okay.
By about 50 million years later, you find bodies of all kinds. Bodies with all different sorts of appendages, antennae and limbs and so forth.
The Cambrian was evolution's big bang. But were toolkit genes the trigger? It may not be that simple. Toolkit genes were probably building simple organisms before the Cambrian, so this potential somehow remained unrealized. Also, there may have been environmental causes. Ancient land masses were changing, riven by inland seas and lakes, expanding ecological niches for animals to differentiate. Perhaps a changing environment pushed the pre-existing genetic toolkit into high gear.
At some point, you got the tools together where all of a sudden there was a quantum leap in the ability to suddenly make new types of bodies.
And among all the new types of bodies, our branch of the tree started too.
So this is one of our oldest ancestors.
Well, it's related to it. And in fact, you know, it shows the basic body plan that we have today. It has a nerve cord that runs along the top of the body. It even has gill slits. This little worm contains a piece of our own, essential parts of our own body. And it appeared about 530 million years ago.
Eons ago, an ancient worm launched our basic body plan. And it would become a predominant way of making bodies. Symmetrical and streamlined, bilateral, and laid out head to tail. Today, most of the animal life on the planet, including humans, is built on this same bilateral design. It's perfect for mobility. Over time, it's given rise to a huge range of successful survivors. And the genetic toolkit has traveled up the evolutionary tree with them.
Pull us back downward toward the ground.
For the Spore evolution game, developers soon realized that making creatures bilateral, identical on either side, needed to be hardwired.
That's one of those things that I think you kind of take for granted, the fact that, you know, all the legs have to
move the same distance, no matter how long they are.
Early product testing showed that despite the wild designs players would make, bilateral patterning was almost always a given. Evolution's ancient genetic toolkit laid out a basic bilateral body. Then, a further step expanded the potential for generating new kinds of bodies: segmentation. Will Wright is at another Evo-Devo lab to see toolkit genes in action.
So we want to understand how this incredible, wonderful diversity is generated, what makes these animals different from one another. But to do that, we have to understand how the basic body plan of all of these things is set up.
They all use the same tricks to create this diversity.
Exactly. What we've discovered is that there's the same sort of toolkit of genes that's always used to make the animal.
Once again, the fruit fly is the preferred specimen. Its embryos are obliging to geneticists like Nipam Patel, eager to pop open the hood and check out how it's really built inside.
So we've designed this so that we've been able to detect where the gene is on. What you're going to see is stripes appear on the embryos.
All right.
There we go.
Ooh.
And we can see now very nicely the repeated pattern of segments. So it's a ladder-like pattern with a rung in each segment.
This is basically his spinal column?
That's right. So the whole body is organized in these very nice repeating segments.
It's the groundbreaking step in forming a fly. Just as they did with the ancient Cambrian ancestor, these genes are organizing the body into top and bottom, front and back. But they are also segmenting it into zones of activity, marking positions for organs, appendages, and limbs.
So you have certain genes that are also telling certain segments to specialize. Is that what's causing the brain to occur at one end?
That's right. So eventually, every one of these segments will actually have a unique property to it.
Whether it's in a fly or a mouse, horse or human, similar genes are laying out a body architecture, and segmentation is the method. It's also a moment when the ancient recipe for building bodies can undergo mutation, allowing for accidental genetic creativity. New shapes can come along from DNA malfunctions, causing a toolkit gene to switch on early or late, or never switch on at all.
So could you potentially turn on 2 or 3 more brains in here if you could modify that gene?
So we could do things like that, so that more of the animal could become brain, for example, or do the reverse, and get rid of the brain and turn it in so that it's identical to the abdomen instead.
So you could actually make a really smart fruit fly at this point.
Well, unfortunately, when you do those things, they die.
Oh, I guess that's not really smart.
A mutation causing a fly with 2 brains might not work outside Hollywood, but subtler changes, say a slightly larger wing, or a second set of wings, may have real advantages, and that new design could catch on. Creatures and their body parts aren't invented from scratch. They evolve when their genetic networks are modified by ancient toolkit genes. Evolution of form is a matter of teaching very old genes new tricks.
If you take a look at the difference between a mouse and a cat, the difference is not new types of cells, but change in organization. Somewhere along the line, those instructions were modified, not in a way that scrambled them, but in a way that gave subtle changes to lead to the differences between organisms.
Of course, not every organism can stack up against the environment. The genetic toolkit builds bodies that are, at best, hopeful possibilities. Fossil beds are littered with one-hit wonders, evolutionary experiments that prove to be maladapted dead ends.
Genes propose, and the environment decides. That genome is not a static thing. It's constantly roiling around, changing where genes are turned on and off. And whether these new traits that are constantly being pushed out there, whether they're accepted, depends on the environment. That, baby, is evolution.
Evolution has taken millions of twists and turns. How did toolkit genes that made an ancient worm get reconfigured to eventually make the parts that make us? Our hands, our hearts,